Oh no, it's a plasma membrane. This is our plasma membrane project by Keene
and Gentryplasmamem.
I'm going to introduce all the parts, starting with cholesterol, glycoprotein, glycolipid, integral transport protein, carrier protein, and phospholipid bilayer. And the channel protein. I'm going to start off with cholesterol. Cholesterol doesn't span the whole length of the bilayer. It also stabilizes phospholipids. It plays a role in facilitating cell signaling. Cell signaling is the ability to communicate with other cells. Cholesterol contains both hydrophilic and hydrophobic portions. Without cholesterol, cell membranes would be too fluid, not firm enough.
The phospholipid bilayer is composed of hydrophobic, nonpolar, water-hating tails and hydrophilic, polar, water-loving heads. The tails extend inward and the heads outward. This organization of phospholipids in the plasma membrane make the latter selectively permeable to ions and molecules. Phospholipids provide a barrier around the cell and are also emphatic, meaning they are a single molecule with a dual personality.
Integral protein, aka transport protein, firmly attaches in the plasma membrane with its hydrophobic component interacting with the phospholipids. H2O, aka water, loves to travel through the transport protein at quick speeds, very quick speeds.
The carrier proteins regulate entry and exit of particles into and out of the cell. A carrier protein is required to move particles from areas of low concentration to areas of high concentration. They help in the conduction of ionic substances and polar substances. Each carrier protein is designed to recognize only one substance or one group of very, very similar substances. Specific diseases have been correlated with specific defects in carrier proteins. Carrier proteins may assist movement of substances by facilitated diffusion or active transport, but they do not use the same carrier protein. This is also called the sodium-potassium pump.
Meow. Meow.
Glycolipid.
Glycolipid is a membrane component composed of lipids that are covalently bonded to monosaccharides or polysaccharides. The outer surface of the membrane will tend to have a lot of glycolipids. Glycolipids have their hydrophobic tails embedded in the hydrophobic region of the membrane and their heads exposed outside the cell.
Active transport is the movement across the membrane against the concentration gradient. This requires energy. Examples of active transport include the uptake of glucose in the intestines in humans and the uptake of mineral ions into root hair cells and plants. The sodium-potassium pump carrier protein requires active transport to move the sodium and potassium through the pump. Exocytosis is molecules exiting the cells, and endocytosis is molecules entering cells.
Passive transport. Passive transport is moving particles from one high to low concentration and does not require energy. Passive transport is dependent on permeability of the cell membrane. Four main kinds of passive transport are diffusion, facilitated diffusion, filtration, and osmosis.
Facilitated diffusion is the passage of molecules and ions across a biological membrane through specific carrier proteins and require no energy, used especially in the case of large polar molecules and charged ions.
Osmosis, the diffusion of water molecules across a selective permeable membrane. The movement of water molecules from a high solution of water potential to an area of low water potential. Diffusion, O2 and CO2. It can happen without a cell membrane. Diffusion is the movement from where there's a lot to where there's not. Move straight across that plasma membrane without a protein. Hey, it's a plasma membrane! This is our phospholipid project with approach. Restart.